Cecilia. I'm a postdoctoral researcher at UK, University College London. I am originally from Argentina, and today I will talk with you about this project where we use these single molecule techniques to study a specific protein of the HIV virus. So first of all, I will discuss a little bit the techniques that we use, and then I will go into the description of the system. Okay, just to give you a little bit of concept, the typical scale, if you want to study tissue, animals, or single cell of bacteria, you are okay using typical wide field or confocal microscope. But if you want to study some viruses or proteins in fluorescence microscopy, you need to use different kinds of microscopy. The types of microscopy that we call super resolution microscopy, this will be STED, STORM, or what we call single molecule localization microscopy, and these are the techniques that we use in our lab to study single proteins or even single DNAs. Okay. So, in these techniques, you use a fluorescence microscope. You study dyes. The size of the dyes that you have is, for example, for ATO679, it's 1 nanometer, and if you image this dye in a camera or your sensor of a confocal microscope, you will see something like this. This is the image of the single molecule, and it has 200 nanometers in size, so 2 orders of magnitude bigger than the actual labeling of what you are studying. But what happens if I ask you, okay, if you have this single molecule, where is the center of this single molecule? I hope you say, okay, it's more or less here, right, in the center of this. So we can model the image of the single molecule with it's called the Airy disk function. It's super similar to a Gaussian, so we can fit this with a Gaussian, and we can estimate the center of this Gaussian with a localization precision of 20 nanometers. So we go from an image of 200 up to 20 nanometers. So the key of these techniques is having single molecules, so you can fit them and have a really small localization precision. For having single molecules, what you have to do is separate them, right? Because typically you cannot resolve them. So what we do to separate them, we separate them in time. So to this, we add a blinking mechanism where we can turn on and off our dyes, and we can separate them in time. So imagine here you have this smiley face structure that is diffracted limited. If you want to check if you have all the dyes, all your proteins labeled with proteins, and you do an image, you will see something like this, and you don't have any detail. You cannot see if it's a smiley or sad face. But instead, if you can turn off all the dyes that are labeling your proteins, for example here, and you can turn on only 3 of them, you can localize them. You can put a cross where you can find them. You turn them off. You turn them on. Different ones, you localize them, and in the end of your experiment, you will have the localizations of all of them, and you will have a map of localizations. So the final figure that you will see here will be a map of localization. So all the figures that I will show you from now on will be a map of localizations, no exact images from a sensor, okay? So this is an example where you have this, could be a nuclear pore complex, for example, where you have the blinking dyes, and at the end of the experiment, you analyze this, you localize all the molecules, and you can have, for example, millions of these single molecules, and at the end of the experiment, you find something like this, and you can see that you have a hole, you can measure this, and many more things. Okay. So how we perform this blinking mechanism. The blinking mechanism will define the name of the technique that you have. It could be STORM, PALM, or PAINT. And in our case, we use DNA PAINT. Let me go. Yeah, we use, DNA PAINT, where we label our antibody or protein or what we want to know with a DNA docking strand, and this is like around 10 to 7 bases. And in the buffer, we add a solution with the complementary DNA strand that has a dye, and we can use any dye. Nowadays in the community, we are using, all of us, Cy3b, and we can use this with Fab fragments and also nanobodies. And now it's really useful to use these alpha tag, spike catchers, or halo tags because they are smaller. since I told you before, the resolution that we're getting with this technique is around 10 nanometers. So if you have an antibody that is 15 nanometers, it's like competing with the resolution that you have. So the idea is to have the smaller tags that you have to get better resolution. And more important things, we also can have, we can label this, we can have, anti-GFP and anti-M-cherry nanobodies. So if you already have a structure or cell lines that are expressing GFP, M-cherry, M-scarlet, you can also use this technique with this. So the blinking mechanism that we use is the binding and unbinding of a single DNA strand to a docking strand that is targeting our antibody or protein, okay? Okay, so another thing that we can do is like we can do some quantitative measurements. Imagine if I have one protein labeled with one antibody, I know I can measure the rate of binding and unbinding. Imagine it's 500 milliseconds. But instead, if I have one, instead of having one, I have two of them, the time that I have to wait for the other DNA to come will be half of that. And if I have four of them, I will have to wait half of the time. So measuring this rate, what we can do is like measuring how many proteins we have in our cluster of our diffracted limiter size. So we can do a calibration. If we know the rate of binding and unbinding of one protein, we can measure and have the number of oligomerizations in our system. So also these techniques, we can combine them with multiplexing. We can label different structures with orthogonal DNA base. For example, here I'm showing an image of nuclear pore complex, microtubules, and mitochondria. We do some exchange, and you can have this beautiful three-color image, and this has been done in a commercial confocal microscope, specifically a spinning disk microscope. And also in 2023, they published a technique where you can reach Armstrong resolution also with these techniques. And here I'm showing you an image of nuclear pore complexes where you can have like 0.5 nanometers localization precision. Okay, and this is the distance of two of the pairs proteins of the nuclear pore complex. But going back to the viruses, just to give you an idea of the scale of the different viruses that are there, if you want to study Ebola virus, you are good with a confocal microscope, but then if you want to have some details of the coronavirus or HIV in our case, or a dengue virus, you need to go down and use these super resolution techniques, okay? So HIV, it's, I will treat it like as a sphere of 100 nanometers, which is decorated with these proteins, what we call envelope glycoprotein, which is a trimer formed by 3 GP120 proteins, and the gag that is attaching to the membrane to the virus. And more or less, you have around between 15 to 20 of these envelope glycoproteins around this sphere, okay? And this, we are really interested in this protein because it's the first step of infection, because attached to the CD4 receptors of our immune cells, okay? And this virus, after assembly in the host cells, goes from an immature state to a mature state, and only the mature states can infect the next cell, okay? So these viruses has been studied using cryo-electron microscopy. Here I'm showing you some of them, and I hope you can see the decoration of this M protein around them. And this is giving us really high resolution, but the counterpart is you need the virus is vitrified, and also this hardly has been seen in mutants. Nobody has been seen this in the native virus. On the other hand, you can use these super resolution techniques. You don't reach the same resolution as cryo-electron microscopy, but you can do these images in a biological environment. So in our case, what we have, first of all, we have a biosensor to detect between this mature and immature system. We have a GFP attached to the gag protein of the membrane of the virus, and we add this acceptor called fast. When both are parallel, we have a FREC signal, and then when the virus go from the immature to the mature system, they change the conformation and there's no more FRET. And this changed the lifetime of the acceptor, so we can measure this difference in the lifetime, so we can say if we are in the mature or immature, we have the mature or immature system. Here I'm showing you some examples of the mature and adding the protease to transform this to the mature and checking the lifetime. We can check that we have all of them in the mature or immature system. And also we perform infectivity assays to check the maturation of the virus. Okay, as I told you before, we would like to have really good resolution, and antibodies are really big compared, and actually the size of the antibodies are compared to the protein that we want to study. So we say, okay, what we can do to get a better resolution, more detail on this system. So we say, okay, what about if we add an alpha tag? Why the alpha tag? Because alpha tag is the best that you can have now. It's the smallest tag available now in the community. And also it has a really good label efficiency, around 45%. You can have with antibodies 20%, but this is around 45%. So we use AlphaFold to try to figure out where to add an alpha tag and do some super resolution imaging on those virus. So for this part, we find 26 options to put the alpha tag in the before loop. This envelope glycoprotein has many loops, but not all of them maintain the infectivity, and not all of them are exposed to be able to attach to the DNAs that we will have later. So we find 26 options, and for all 26 options, we ask AlphaFold to give us 5 structures. And then with these 5 structures, we check for malformed tags, and we check for the changes in the conformational of the host. And then with all of these possibilities, we got 4 of them, and what we did is like we create these 4 versions, and we check the protein expression, the viral infectivity, and we did some cell-to-cell fusion assays for all these 4 options, and we keep the best of them. For example, here I'm showing some examples of what we check. Here we have the GAG inside the membrane, and also the alpha in the envelope glycoprotein. So we did some Fluorescence correlation microscopy. In violet, you can see the GAG, and in orange, the signal of the envelope glycoprotein. And you can see that if there is no alpha, they don't correlate. This is the cross correlation in blue. And when we add the alpha, they start to do some correlation. And this is one example of one of the cases that AlphaFold gave us, and it didn't work, so you cannot see any correlation between them. So this is one of the tests that we did to check if everything was working well. Okay, so our system is something like this. We have our sphere decorated with the envelope glycoprotein. We have each of them of the GP120 labeled with an alpha tag. Then we add an alpha nanobody with the DNA strand, and in solution, we have the complementary DNA strand binding and unbinding, so we'll have our single molecules. So we go from an image something like this of the system to something like this, where you can see that we can measure this. We have a sphere, all the M is not all around, it's only forming like an equator in the system, and you can see that we have more protein on this side in the specificals than this size. And in this case, we use site 3B. Just to give an example. So what we did is we did some min-flux imaging of this. This is a technique that can give you 5 nanometer isotopic resolution. Each of these blobs represents 1 M, and so we did this 3D resolution. And then using DNA paint, we have all these examples. Here I'm showing you with the circle the surface sphere of the virus, and you can see that the envelope glycoprotein is forming some clusters here, over here, and some of them, I hope you can see that they form like a small triangle here and here. And here I'm showing you some zoom views of these triangles here and here. And here, this is the expected structure of one of the mutants, so they are pretty similar. And here I'm adding the guide to see these triangles. We are really at the limit of the resolution of the techniques, but I hope you also can see the triangles here. So here are more examples of the mature version of the virus. Here you have some clusterization here, and some of them are forming only 2 to 3 clusters. and here are some examples of the immature version of the virus. So with all of this, we did some quantification of this, so we can check, we separate them in 3 cases, one forming 1 cluster, 2 clusters, and then 3 or more, and we perform this for the wild type. We are adding or not adding CD4 for the mature and immature, and also performing the delta CT, which is adding some modification to the GAG protein. And with this, since we can measure and quantify the amount of proteins, what we did is like we count how many proteins we have using QPaint, the technique that I showed you before, and more or less we have in average 20 envelope-like proteins per virus, okay? So with that, I would like to conclude, just to tell you that I show you that we can use these techniques, super resolution techniques, and this has been the highest resolution in viruses that I can tell for now using the fluorescence microscopy. I show you how we use AlphaFold to tag our viruses, and with them, we show that we can do super resolution on them. I show you how we do, I didn't show you the single molecule thread, but I show you the fluorescence correlation microscopy that we did for checking the functionality. And in the future, we're interested in checking the close conformation of the trimer, because the trimer can go from a close conformation that nobody has seen to an open conformation. So we're interested to see this to complement cryo-electron microscopy techniques, cryo-electron microscopy images that we have. And this is something that the super resolution community is going now, like trying to complement structural measurements with fluorescence measurements and have an idea of the conformational changes. And with that, I would like to thank my lab, my supervisor, Sabrina Simoncelli, and the Padillas lab in King's College London, where they help us with the viruses and all of this, and all of you for listening to me.